Welcome to my channel. Today we are going to study about an organism which is small enough to be overlooked, powerful enough to support ecosystems, nutrition, and science. Yes, it is green algae. The tiny organisms with a huge role in oxygen production, protein supply, and biotechnology. Watch this video to updated information about some species of green algae. This video is going to be very interesting and helpful for you. So, it is more important for you to subscribe this channel to get new videos on time. So, press the bell icon below this video. Also comment and like. And share this video with your fellows. Today's video is about production of green algae like Gelidium amansi, Gracilaria species and Eucheuma cottoni. Terms related to life cycle of seaweeds will also be discussed. Major pests and competitors of seaweeds and seaweed cultivation practices in Pakistan. Let's discuss the Gelidium amansi. This weed is commonly cultivated in Japan for obtaining agaros. Cuttings of the plants are sown in protected base, where they generate new fronds. Horizontal, vertical and both types of rope raft cultivation techniques similar to those used for Undaria are presently employed for cultivation of Gelidium amansi. Their growth is enhanced by using fertilizer pellets. The genus of Gracilaria is cosmopolitan in distribution and has been reported from the Arctic, temperate and tropical regions. Erect thallus arise from a small discoid holdfast. The thalli are generally cylindrical, depressed or blade-shaped, with lateral, alternate or subdichotomous branches. Sometimes several different branches may be found in one plant. The external appearance of thalli may be used to identify species. The style of the apex and the base of branches are different with species. Gracilaria confervoides and Gracilaria gigas are commonly cultivated in various parts of the world, especially in Taiwan. This weed is commonly cultivated in ponds used for culture of milkfish. This species can adapt to wide variations in environmental conditions like salinity ranging from 8 to 25 ppt and high temperature up to 25 degrees centigrade. Cuttings are planted uniformly on the pond bottom at the rate of 3,000 to 5,000 cuttings per hectare of pond. These cuttings are supported by bamboo sticks that are dug into the bottom of ocean. The depth of water in the pond increased up to 60 to 80 cm with a rise in temperature in June. Exchange of tidal water is maintained to provide additional nutrients and to adjust salinity level. Let's discuss the life cycle of Gracilaria species. Mature tetrasporophytes, 2N, produce meiosis, tetraspores, some of which develop to male gametophytes, N, while others grow to female gametophytes. The male mature gametophyte forms spermatangia, the female forms carpogonia. After fertilization and cystocarps are formed on the female plant, carpospores, 2N, are released and develop to tetrasporophytes again. The vegetative tetrasporophyte, male gametophyte and female gametophyte have no obvious difference. Wild Gracilaria collected consist of tetrasporophytes, spermophytes, and carposporophytes in different stages, respectively. Let's study some very important terms related to seaweed life cycle. Carpospore, a non-motile, diploid, 2N, spore of red produced by the carposporophyte. Carpospores develop into tetrasporophytes. Carpogonium, structure on a female gametophyte that produces gametes. Carposporangium, the cell in a carposporophyte that produces carpospores. Carposporophyte, formed by the union of haploid gametes, diploid, 2N, stage of the life cycle that is carried by the haploid female gametophyte in the cystocarp, produces carpospores. Cystocarp, structure produced on female plant after fertilization, houses the carposporophyte. Spermatia, non-motile male gametes. Spermatangium, structure on a male gametophyte that produce spermatia, male gametes. Tetrasporophyte, free-living, diploid stage of the life cycle that produces tetraspores by meiosis. Tetrasporangium, a spore-generating structure in the tetrasporophyte phase of the life cycle that produces 4 haploid spores. Tetraspore, one of 4 haploid spores produced by meiosis in a tetrasporangium that develops into a gametophyte. Gametophyte, haploid plant that develops from a spore, produces gametes. Fertilization of the water body can be done through inorganic or organic fertilizers. These fertilizers are applied at the rate of about 3 kg per ha of urea every week or 120-180 kg ha of fermented pig manure every 2 or 3 days at times of water exchange. If the water temperature falls below 8 degrees centigrade, either the depth of the pond water is increased or the plants are transferred to protected wintering ponds. Do you know about the major pests and competitors of this seaweed? Let's see. Some other algal species like Enteromorpha and Chaetomorpha have ability to overgrow and occupy the area. So these species compete for space, shelter and nutrients. To control these species, following measures can be adopted. Lower the water level. Reduce the water exchange. Plant additional large gracilaria to utilize nutrients in the pond water. Stock adult milkfish or tilapia of about 150 gram weight at the rate of 500 to 1000 specimens per ha to feed on the green algae. This species can be harvested when mature. Harvesting of gracilaria from the ponds is done by hand or through scoop net starting from June to November. The annual yield is around 10 tons hectare. The main costs of production are used for labor, which is about 53% of the total cost, while cost of seed is about 18% of the total cost. The rate of return on initial investment is 50%. The annual average yield is around 10 tons hectare. This seaweed can be polycultured with crabs. Through polyculture, production of gracilaria is reduced to about 9 tons hectare. The operating costs increased more than 4 times. But an additional production of 6.3 tons ha of crabs and shrimps is obtained. The harvested plants are sun-dried on for export bamboo screens and plastic sheets. That's all about gracilaria. Let's study Eucheuma species, which is a red algae. Eucheuma species. Commercial operation in the Philippines for production of Kappa carrageenan. The most successful species so far is Eucheuma cottony. But the related species Eucheuma muricatum is equal to spinosum has also been tried. Eucheuma cottony is shown in the picture. The off-bottom method uses stakes which hold the ropes with the propagules attached and must be done in shallow and clean water. Rafts are frequently used in deeper waters but require boats to go to the farms. The structure to keep the ropes inside the water is provided by rafts made from bamboo, wood or PVC pipe instead of stakes. Long lines can be used as in off-bottom, as in the raft methods, and is a more intensive cultivation process. It is recommended when large areas are being used for the culture of E. cottony, protected coral reef areas with a good water flow. Temperatures between 26 and 32 degrees centigrades are selected. Nets and long lines are used for suspended culture. Major factor in the success of commercial culture of Eucheuma cottony in the Philippines is the use of a clone of the species known as tambalang, produced by natural selection. It grows and multiplies rapidly. It can survive in a wide variety of environmental conditions. Vegetative parts of the plant are attached to polyethylene nets strung parallel to the bottom between poles. The seed is attached with thin plastic strips which allow movement. Do not cut the fronds. When the fronds reach a weight of 1200 to 1500 grams, harvesting is done by pruning about 1/3. Further harvesting is done when the fronds grow to the desired size, and this cycle is repeated. Long lines are preferred to nets for grow-out as they are easy to maintain, although the productivity per unit area may be lower. The farming of the seaweed cottony, Eucheuma striatum, began in the Philippines in 1960s under the auspices of the University of Hawaii, UZ and Marine Colloids, a U.S. carrageenan firm. However, mariculture started seriously only in 1973. when a cultivar was discovered by a Sitankhe farmer called Tambalang. This variety was later recognized as a totally different species and genus and renamed Capophycus alvarezi var. Tambalang in honor of Mr. Vicente Alvarez, a biologist of the Bureau of Fisheries and Aquatic Resources who became the first manager of marine colloids, and Mr. Tambalang, the true pioneers of cottony cultivation. Experimental culture of several other species of seaweeds is being carried out by many institutions and private enterprises. Chondrus crispus, Irish moss, is cultivated in greenhouse tanks flushed with seawater. Eucuma isoform is cultured in tanks with slanted bottoms in circulating water to keep the plants in suspension. Mass culture techniques using sporophytes as seed have been developed for enhancing natural stocks of the kelp macrocystis. Seaweed cultivation in Pakistan. The macrobenthic algae are an important component of the marine ecosystem. They exhibit an enormous variety in size, color, form, and lifestyle and are extremely rich in the sea. The seaweed resources are abundant around the rocky shores of Karachi, the coast of Lasbela. Standing crop of intertidal seaweeds from the Buleji and Manora coast of Karachi. Recently natural bed of seaweed at Paradise Point, Karachi coast. Pakistan is a developing country. The marine resources like seaweeds are under threat due to variety of pollutants. There are above 6,000 industries located in Karachi at different sites. According to an estimate, all these industries contribute approximately 99% of the total industrial pollution beside the contamination from oil waste discharge. At Nathia Gali of Karachi coast no any information on diversity of seaweeds, although this coast has very luxuriant seaweed vegetation. Therefore the present investigation deals with the seaweed's biomass, distribution, diversity of seaweed species, Chlorophyta, Phaeophyta and Rhodophyta, of Nathia Gali coast, Karachi, Northern Arabian Sea. Thanks for watching. Like and share this video. Subscribe this channel to get more informative videos and to become part of a growing scientific community.